Welcome, everyone. We are going into chapter 16, amino acids, proteins, and enzymes. This is GOBC Chem 114. I hope you're ready for the last part of the course, one of the most important parts of it, putting it all together. Before we get started, though, thinking about the possible opportunities that exist for people in the health professions, certainly physicians' assistants roles, some nursing assistants, nursing positions, very important. Everybody is needed for the healthcare system. There are so many roles to fill. Um, uh, Things such as patient medical records and history, diagnosing illnesses, educating and counseling patients about their options, and referring the patients to specialists are some of the things that physician assistants can do when they don't have all the answers. No one has all the answers. We just do the best we can and we get help. That's the real world. And so we look at proteins. They're made up of amino acids, and so we're going to learn about amino acids and how different they are, and they're monomers, and they make up we make up dimers and tripeptides and tetrapeptides and polypeptides, and then proteins. Eventually, when we get past 50 amino acids, we actually have an enzyme, which is the bare minimum. But even smaller than 50 amino acids, we can have uh, effects from four or five peptides that actually play a role in our body, such as we're going to learn a little bit about that as painkillers. So, five amino acids put together, minimum, sometimes can have an effect on the body that's quite profound. Looking at this here, proteins, they make up the horn of this animal, if you can imagine. Look how strong and powerful that is just from a protein. And proteins can make up our hair, our skin, our fingernails. So proteins are pretty important structurally. We have collagen and all sorts of stuff in our knees and our bone marrow, just amazing. And so we have 20 different amino acids really to think about. I don't expect you to memorize all those at this point. If you take another biochemistry class, you may need to memorize that. There are some similarities for every amino acid, but there are some differences, and those differences are from the R group. Okay, every amino acid is going to have an amino terminal. Every amino acid is going to have a carboxylic acid part at the end and an alpha carbon. Um, the structural components for certain proteins are cartilage, muscles, hair, and nails. They function as enzymes to regulate biological reactions such as digestion and cellular metabolism. So enzymes are working all the time in our body, right? There's millions, maybe billions, I don't know what the number is, but we have enzymes everywhere in the human body. We have also other functions of proteins, not just as enzymes, but they can be carriers such as hemoglobin and myoglobin, which carry oxygen and iron and CO2 and all these cool things, such as transferrin, which is a transporter of iron. Furan, beginning with an F, as we know, iron is also called, and we'll learn about ferritin a little bit. And speaking of these examples of proteins, structural classifications of proteins, we see here 7 different classes. We have structural, contractile, transport, storage, hormone, enzyme, and protection. And so some that provide structural components of for our human body would be collagen for our tendons and cartilage and keratin in our hair and skin. We have contractile proteins, which are responsible, they're called myosin and actin, to help contract muscle fibers. We have transport proteins that I just mentioned, such as hemoglobin, and lipoproteins that transport lipids. We looked at chylomicrons earlier. We have storage proteins. Those can be casein, that stores protein in milk. You did an experiment in that this semester. Ferritin, which stores iron in the spleen and liver, contains a lot of iron. Iron is needed for enzymes and to do the chemical activity, what they call enzymatic activity. So you can move that ferritin, another protein supporting another protein. Ferritin stores iron, and iron could be used by an enzyme to allow it to be active, to catalyze a biochemical reaction. But before that, let's talk about hormones. Insulin regulates blood glucose levels, and growth hormones regulate body growth. So that's another protein. What about enzymes, right? Uh, so, so, so hormones can be proteins and they can be steroids, right? We've learned that. Enzymes catalyze biochemical reactions in the cell. Sucrase catalyzes a hydrolysis of sucrose. Trypsin catalyzes a hydrolysis of proteins. So if we needed to break down a protein, we have another protein to do that. Wow. So this is just crazy how this is coming together. Protection, we have immunoglobulins, immune responses. Those are proteins that go after bacteria and foreign viruses and bacteria as some protection in our environment. Amino acids, here the bulwark of all this. What we've been talking about, what's so important, leading to this big should I say, big ending, the apex of our learning in this class is learning about amino acids, and we learned about how this amine can be a base, and this carboxylate can be a carboxylic acid. But we're in 7.4 media, so this is what the backbone, the blue and the pink are the backbone of every amino acid, and as well as the alpha carbon, which can be a little bit different depending on the structure of the amino acid. But the R group, the yellow, is the surprise. That's what varies in each different type of amino acid. The nature for us humans requires 20 amino acids. We don't have all of them manufactured in our body. We do make some amino acids. Others are essential amino acids we have to go out and get through plants and animals. These are the molecular building blocks of proteins. Every amino acid has an alpha carbon. Every amino acid has an ammonia group NH3 plus. Every amino acid has a carboxylate, the COO minus. Okay, so we know this. And here is our ball and stick model to kind of help us see this a little bit better than the two-dimensional version. So we can start to see there's twists and turns in these amino acids. Remember that when we get to proteins. Amino acids are classified as nonpolar with a hydrocarbon side tail. That would be this valine. We have polar molecules that have ionic side chains right here that have polar bonds, and they have this nitrogen, which can hydrogen bond. And both of the blue areas are the same, whether it's valine or asparagine, they have the same backbone. It's the same. What differs? The R group, making the now you might say to yourself, but Mr. Pryor, this is positive and negative. This is positive and negative. It's got to be water soluble. You're correct. But when you start putting it into a protein in a long chain of these connected together through what? The amide bond. Don't forget the amide bond because that's what makes up a peptide bond, right? That's why we learned about amides, how amines and carboxylic acids could react and you get water and you get an amide bond. Well, that's what you get when these amino acids come together. And enzymes do this. We're not doing it in a lab, we're doing it in vivo. Enzymes actually can do this. Here's some more nonpolar amino acids. They're hydrophobic. Again, glycine with just a hydrogen, alanine with a methyl group, valine with this kind of like an isopropyl like alkyl group. Um, then we have a leucine, which is kind of got an extra carbon thrown in here compared to valine. Then isoleucine, and we have again 4 carbons arranged a little bit differently. And so all these amino acids, they have abbreviations. For glycine, it's gly, and alanine, it's ALA, and valine, it's just VAL, and leucine, LEU, and isoleucine is II or ILE. And then you have single letters of the alphabet that can be used to describe amino acids too. Now, moving on, we see phenylalanine right here I'm pointing to that has a benzyl group. We see methionine, which has a methyl group in the middle of 2 carbons. That's a little bit different. We have proline here that's kind of a circle instead of an open free chain of any type, which helps with making turns in proteins, making the protein bend. We got tryptophan, which is often a base, it's a proton acceptor and a proton donor, depending on the environment. Tryptophan, TRPW. So these, you can go through this. PHE, which is F for phenylalanine. So some of the letters don't always match the name, but most of them do. Methionine is MET, MET. Proline is PROP, and tryptophan, TRP, but it has a W, for example, as the letter representing it. So these are the structures and how each monomer or each amino acid looks when the pH is 7.4. The ammonium group is protonated, the carboxylic acid group is deprotonated. We have polar neutral amino acids. What the heck is that? Well, you see there's really no charges on here, but they are polar because you can get some hydrogen bonding here. You have some alcohol groups, a thiol group, an amide link, a amide here, yeah, an amide and another amide here. So you have serine, which is SERS. You can see the alcohol group. You have threonine. You have THRT. You have tyrosine, which is TYR, which is abbreviated with a Y as well. That's a benzyl group with a hydroxyl, which is called a phenyl group, and then a methylene group here. Then we have the thiol group with a methylene group. We have the amide and the methylene, a couple of methylenes here, and another amide linkage. And so, which was glutamine GLNQ, asparagine ASNN, cysteine, which is CYSC, involved in disulfide linkages and proteins, tyrosine, TYRY. It often deprotonated and protonated phenol is useful in enzyme pockets. Enzyme pockets are sites of enzymatic activity where substrate can be bound to the enzyme. The substrate of an enzyme is the molecule or molecules that that enzyme can bind and do chemical reactivity to, okay, in simplistic terms. So we see it here, polar neutral amino acids. Everything in blue here is typical, and the charges disappear if you link all these together except for the beginning and the ending charge. But we have to go by the R group when we look at them as monomers to determine what type of amino acid we're dealing with. These are hydrophilic and they're polar. Polar acidic amino acids, what does this mean? Well, now they have charges on them. And we have aspartate, which is ASPD, and then we have glutamate, which is GLUE. And so these are very important in the cell. We'll see, you can see that if you take more biochemistry, you will learn about how they can be active in enzyme pockets. Histidine, lysine, and arginine are polar basic amino acids. They're hydrophilic. Some of them have charges, all of them do in this case, where they can accept a proton and even donate a proton depending on the enzyme pocket in which it's located in. You can look at the abbreviations: histidine, HISH, lysine, LYSK, arginine, ARGR. So remember, the one letter alphabet soup used, sometimes it's not exactly like the name. Arginine, R, that's close. Lysine is K. There's no K even in lysine. But yeah, so you should have an idea about these classes of different amino acids as you move on. We already discussed the ammonium group. We have the carboxylate group. We have the alpha carbon, and then we have the R group. Okay, so that is just a review. Drawing these out may be important in some classes. Limited utility here as I introduce you to these. I don't really expect you to be able to write everything out, but being able to draw out an amino acid at this stage should be fairly easy. Not that you're going to fail this class if you can't draw out an amino acid. I wouldn't do that to you at this stage, but you should kind of know how to draw this backbone out. And well, what are we looking at here? Well, is this polar or a non-polar amino acid? Well, don't get confused with the positive ammonium group and the negative carboxylate. That's on every amino acid. We know that A as glycine, it just has a hydrogen. So that's going to be non-polar. In the protein, we got to think about the amino acid being translocated to a long protein, and what exactly would that R group mean in that context, not in the context of the N terminus and the C terminus. We look at threonine, we see a hydroxyl group here, a methide and a methyl group. So guess what? We have, this is going to be polar. It's going to dissolve in water with respect to the R group. Okay. So nonpolar for glycine, polar for 309. Hey, can you draw these out? Valine and cysteine. Well, can you look them up efficiently is one thing that might be helpful. And if we move on, valine, we're able to draw this out. We draw valine, that's completely hydrophobic with respect to the R group. Cysteine, it is SHCH2, that is going to be polar, right? So, and that is abbreviated CYS or C. Valine is VAL or V. And that does it. You guys have been awesome. I'll be back with 16.2 real soon. Talk to you later.